bacterial motility, and basically anaerobic growth, as a way you could summarize today's lab. And so for motility, we have two ways to check for this. We could do a wet mount or a hang drop and visualize it under the microscope. And as I said to you guys already, I'm not a big fan of the wet mount because you can get a receding water line. And if you don't recognize that, you may record false results. Hang drop, as you saw, is a little bit more technically challenging, but it's worth it in the long run. And then I don't have you guys do it again because these are techniques you'd have to practice over and over again to really get good. So instead of individually helping each one of you guys, which would take up all of the lab period, I just really do it as confirmation for those of you guys that really need to see it under the microscope. So like for Ashley's, she would have had false negative results, right, had we just looked at her stab. Does that make sense to you guys? And then, how do they move? So this is what Jessica asked, was like, well, how are they moving, right? Well, they move by flagella, especially the ones that we're working with, because we don't work with any spirochetes, spirochetes are the only ones that corkscrew through their environment. Spirilium a lot of times move, but again, they move by flagella. But we don't work with any of those, right? They're too hard to grow in the lab. So you only have two choices, rods and cocci. And for the most part, if you think about a boat, would you want a round boat or a rod-shaped boat? A rod-shaped boat makes sense. So it makes sense, right, that rod-shaped ones tend to be modal a lot. Where imagine trying to put a flagella or a motor on a round boat. You'd just be going around. But there are some that are cocci that do have flagella in our modal, but it's rare, right? So if you have a cocci, it's probably non-modal, right? If you have a rod, there's a possibility that it is modal. And they can have multiple flagella too, not just one. And so we'll talk about the terminology related to that. And that's the only thing really in this other exercise that we have. We don't do the staining again because it's kind of difficult. I actually bought the stain and tried to do it this summer with the STEM kids, and we didn't get any good slides, and I was really bummed. But in trying to figure out the problem with our goggles, I had a drawer over there I was going to use, and I forgot there was a whole stash of slides in it that were from other labs and other people over the years. And so I went through that, and I found a bunch of microbiology slides and a bunch that have flagella stains on them, but I haven't got a chance to look at them. So hopefully, when I get a chance to look at them, we can add them in, and you guys can take a look at these prepared slides where you can actually see the flagella stained. The flagella are teeny tiny, and you have to do a special staining procedure to make them appear thicker than they actually are to be able to observe them under the light microscope. So I've got some potential slides. I had a really nice slide, and it walked away, and I was really upset. But I've got some more that hopefully might still be good. So just hadn't had a chance to take a look at them. We just referred to that one as for reference. We're not going to actually, like I said, hold you to looking at those slides. And then fluothioglycolate we just looked at. And again, the question we're asking is, can your organism grow anaerobically? And then the anaerobic jar, which is this. We don't do this activity anymore because, believe it or not, one of these jars is a couple $100. And we don't have enough for all the classes that work. Some of the seals don't stay sealed anymore for these jars. With them keep cutting our funding, I don't ever see us getting any more of these jars. But the FTM can give us that answer anyways. And I've taken pictures when we've done this one in the past that I have put in this. So we just discuss this one and have you guys read about it, but we don't actually do it. But you are still responsible for the information as it relates to this exercise, this experiment. So the terminology for the different numbers and arrangements of flagella are these terms here that you guys do need to know. But remember, I post my PowerPoint for you guys, so you'll have this. You don't have to scribble it down right now, right? So, and if you're in lecture, you should already know it, right? We've covered this already. So monotrichous, trichous meaning hair-like, or polar, meaning that it's at the pole, is how many flagella and where are they? 1. Mono is 1, and where is it? At the end, right? At one of the ends of the organism, which makes sense too when you're talking about a rod-shaped bacteria. It can be at the end, right, of the organism. So Amphitrichus, amphi means opposite. So there's one at either end of the bacterium. Lopho means a clustering. So Lophotrichus is where you have more than one flagella clustered at one end, or at both ends, right? So, yes. Oh no, a single one? No. No. There, no. It'd be a cluster at both ends, or a cluster at just one end. Would be Lopho, because what Lopho means is clustering at an end. Peritrichus is my favorite. Peri meaning many, and these guys are just completely covered the entire surface with flagella, like scary cousin it, right, from the Addams family. So this one would be Peritrichus, so it's completely covered in flagella. This one's a single flagella at one end, so this one is mono. And these guys are really good at running straight. These guys are really good at tumbling around. But they can run pretty straight too. But as you saw, that one that we looked at under the microscope, I'm pretty sure that one's Peritrichus. And again, it kind of tumbles around. The second one we looked at, I definitely know is Peritrichus. And so for your guys, if you want to know, once you figure out what your organism is, you can find this information on the internet. You know, how many flagella it has and where they're arranged. So what's the shape of these two? Rods. So guess what? Yours is probably either mono or Peritrichus, right, for the ones that we have in this class. Huh?
Is that related to trichomonas?
Trick means hair-like. So that's where the trick is coming in. That's similar. Because they have flagella. Trichomonas have flagella. So the trick is hair-like, right? That term means hair-like. So these guys are what shape? Spirillum. They're not spirochetes, right? They're spirillum. Spirochetes are helical. It's a much tighter turn than you would see here. And again, nobody has this, right, in this class. But if you did, and it had a cluster at one end, or in this case, both ends, this would be Lopho. Lopho is the clustering. One at either end is this one, which is the Amphitrichus. Okay? So again, you won't have to look at slides and identify this. It would be pictures, or you'd have to just know the terms definition-wise. So again, wet mount versus hang drop. I prefer the hang drop. A little more work, but I find it's worth it. And part of it relates to some of the complications you can run into that would give you false results. So if you overcrowd, and we talked about this, how I didn't want to put too much bacteria in that drop of water. If it's a motile organism, can crowded ones move? No. So you wouldn't see them move, and you would think it's negative for motility, right? But would this be a true result? No, it would be what we would call a false result. How would you know it was a false result? You might recognize that there's crowding, right? And be like, oh, I need to redo this because they're too crowded, and that could be why they're not moving. If you do it again and they're not all crowded and it's still not moving, then it's probably a non-motile, right? So you have to look for these complications, right, and try to avoid them as best you can. Will crowding make a non-motile move? No. So would this affect your results? No. If you truly have a non-motile and you crowded it, and you didn't notice it, you got lucky, right? Because this wouldn't have an effect. What about if the cell's attached to the slide? Is this going to impede them from moving? Possibly, right? And so therefore you could end up with another false negative result, something we definitely don't want. In the videos I have posted for you guys in those links, there's one where it's stuck to the slide and it's twirling like this. It's really cool. But, you know, again, it was still moving, though. But the reason why it's moving like that, it's stuck. It's stuck to the slide. So, it's kind of cool. Would this cause non-motiles to move if they're stuck to the slide? No. Right, so this would have no effect. A receding water line. How do you recognize this? As I said, it's the water that's moving. So anything in the water is also going to move, like logs floating down a river, right? Everything moves in the same direction and really fast. So when I squished that one earlier, right, I was like, oh, I got a receding water line because everything was moving fast and all in one direction. Then it's the water settled, and then I could see individual cells moving. So if you don't recognize this, is that a big deal for a modal organism? Not really, because they're going to move anyways, right? So this wouldn't have any effect. But if you've got a non-modal and the water is moving it, you're going to end up with a false positive result in this case, right? Where a negative organism is moving, right? You're going to think it's positive for motility, right? So this is the one you have to really look out for your negatives.
Is that a sensitivity issue?
No. This is just a complication. Okay, if we use an old culture. So one of the problems with the organism that I'm working with, which I can spill the beans with, the first one that I did, which is Ashley's, because I know she already looked at her slides. What does your organism have the ability to do over time? It forms endospores. So I had to make a fresh culture yesterday to make sure I had a lot of living organisms and not spores so that we could see it moving, right? So if we have a fresh culture, right, we're going to have a lot of live cells and they're going to move if it's modal. If we have an old culture, a lot of them could be endospores or even worse, dead, right? So are we going to see more motility then? No, and like the second culture mine I did, I was like, well, this one, it'll be okay because I know it's not a spore form, right? So I know I won't probably have a lot of live ones, but I'll have enough that we will see motility, and we did, right? So if it's a modal organism and it's dead or it's formed spores, is it going to move? No, so you're going to end up with a false negative result, right? If you make the mistake of using an old culture. You want fresh cultures. A non-modal organism. Is this going to affect your results? No, right? Because dead ones don't move. We're not talking about walking dead bacteria, right? No zombie bacteria. They don't move when they're dead. So this wouldn't have any effect. So what do we really have to worry about when we're doing these wet mount type preparations? Is we've got to avoid these falses, right? We don't want to overcrowd. I don't know what you can do about attachment other than say look at a lot of different cells, right? You really can't avoid that. And you definitely can avoid using an old culture, right? Choose to use a fresh culture. And as far as false positives, there is a way to recognize you're seeing that receding water line, right? Which would help you. So like I said, I got lots of videos, even some that I took in lab, different semesters and working with the kids this summer. You guys to see it, but you guys got to see it already, so we'll skip over that. Brownian motion, what is that? Yeah, vibration of the water molecules, and this is affected by what type of energy? Thermal energy, which we refer to as what? Heat, right? Heat. So why do we stain organisms before viewing them under the microscope? To help us visualize them, right? To create contrast. But we can't do that here because what would happen? They would die, right? So as I said, right, we're going to decrease the light to help with visualization. So there's a really scientific explanation of this. I'm going to give you guys a different explanation of this, right? So Peter equates it to pennies, right? But I like to use real-world applications. Imagine what happens when a little kid gets a flashlight for the first time. What do they do to you? Shine it right in your face, right? You're getting blared with the light. And some of us, when we first turn on our microscopes, too, it's blaring, right? All that light coming at you, it's more than you can handle. That's 50 pennies worth of light. You're overwhelmed, right? So can you see the difference between all this bright light in some little organism that's only absorbing a penny's worth of light? No, you're blinded, right? You can't see anything. But if you dim the conditions, right? So if I dim my microscope, so less light is coming through, say 5 pennies worth, can I see the difference between that organism that's only absorbing a single penny and now my background that's only about 4 pennies? Yeah, right? And this is why, too, at night, right, I prefer pitch dark, because then you can't see anything, right? When my husband still has the TV on, the eye mask goes on. I can't take all that ambient light, right? I need to sleep, right? But you know, my son, who's got to have his little bit of a night light, right? I don't like that because you see all those shadows, right? You see those subtle differences between the amounts of light in the room, don't you? Right? You see more things than what's really there because of all the shadows, right? Freaks me out too, right? I believe in ghosts, right? It scares me, okay? So, but we want that type of condition, right? We want that dim light. We want those subtle differences to be accentuated because we don't have the stain to help us out, right? Make sense? Right? All right. So, why do you expect Brownian motion, right? So this is all about heat. Why is this going to increase the longer we leave that slide sitting there? The light, right? And especially these old microscopes have halogen light bulbs. They get hot, right? So the light source is going to heat up that slide, right, and create more vibration. And again, the more work you do with this kind of stuff, the more apt you are to recognize vibration over motility, right? But motility, as you can see, right, it's random all over the place, right, and in different directions, where the vibration is very subtle. Right? And everything's kind of going at the same vibration. It makes me sick to look at stuff vibrating, though. I get motion sick. There's one stain that we used to do at another school using India ink. And so the ink droplets are in the solution or on the slide, and they vibrate under the microscope. I couldn't handle that one. I'm glad we don't do that one here. We use a different procedure. So for the motility test, right, the media we use isn't actually as solid as those agar slants that we use or the agar we're going to use next week in the Petri dishes. And why is that? Why do we don't want it to be solid? Yeah, so they can move, right? So we want separation between the agar molecules so the bacteria can spread out. So in the past, we didn't use the reagent TTC, which when the organisms interact with it, it turns red. So you guys could very easily see your growth in these tubes, even though none of our organisms are red, are they? Right? Some of them are kind of whitish. Some of them are even translucent on the slants. We use this reagent to help us see where the bacteria are growing. It's to help us visualize it. So you can almost say we're staining them. If we didn't use that, usually, especially non-modals, you can see that really well because you will see where they stayed, where you stabbed them in. But for positives, a lot of times they're going to diffuse a lot, right? You guys saw that you had a lot of diffusion, and you were able to see that cloudiness because of the TTC. Where if we didn't have that there, and you weren't a very good observant student, you might not see that your tube is cloudy, right? So what does a positive result look like? Right? It's cloudy. It's almost sometimes even like a tornado where it has spread out all the way around, right? 360 degrees when you turn that tube. They've gone in all directions from the source of where they were stabbed, where you inoculated. So the oxidized soluble form of TTC is colorless. Unfortunately, some of it has been reduced ahead of time in our tubes. Remember, our tubes are a little pinkish? But that's okay, right, because not all of it was. And the rest of it did get reduced by our organisms, and it turned what color? Color. Red, right? And again, really helps us be able to visualize the motility, see where the growth is in the tube. So why is it important that we stab it and remove it, right? Yeah, so that when we are putting it in, it stays in one area. Because if you stab and come out at an angle, right, you just move the bacteria from one place to another in the tube, as you inoculated it. And if you don't know what you're looking for later, you may think that that's motility. But when it's on a single plane like that, it's how you stabbed it, right? So you have to look for that complication. So here, notice it's kind of spread out, right? Some of our organisms, unfortunately, kind of do that are negative, but this is definitely a bad stab, right? They came in and came back out way over here. If you turn this one 180 degrees, you'll see that this is flat. Here is, again, probably looks like they stabbed it twice, right? Or again, stabbed in, moved over, pulled it out. So growth will occur anywhere you stick the bacteria, right? So if you move them as you put them in, then that's going to make it harder for you to interpret. So false positives is where the organism is non-motile, right? But the results look positive. And it has to do, you can have trouble with this if you don't perform the test properly. Right? That's a beautiful stab, right? One single stab. Non-motile organism, it didn't move. This one is that complication that I spoke about, where actually the person did an excellent stab, just the organism kind of leaks into the spaces in the media. So there are two of them that tend to do that. Enterococcus faecalis will sometimes do that, and Klebsiella pneumoniae, which are actually non-motiles. But they'll get that weird effect in the tube where it can be confused as motility. So, false negative. What would it be that you had a motile organism, but yet it looks negative? So, she has a strange complication where I think her bacteria is just too big, right? But what else could stop it from moving? Overcrowding, or not enough bacteria, not enough live bacteria, because remember, only the living ones are going to grow and move. And if the media is made properly. See how this is really light? They probably didn't inoculate enough, right? So if this is a motile, we might not have enough there for you to see enough spreading to get a good positive result. One of the things you could do with this is just incubate it longer. Which is why, again, you guys did these, 2 weeks ago. So at this point, if you got at least some in there, hopefully we'd get enough growth that you'd see motility. So, not inoculating enough live, right? If the media is too solid, not a problem. But also this is why we're not going to use old media, because it will dry out, right, and become more solid over time. Why is it essential that the reduced TTC be insoluble? So let's first talk about solubility. What does soluble mean? Dissolves into solution, right? So I don't know about you guys, but I prefer sweet tea. But if you go to a restaurant, right, and it isn't already made sweet, I say no thank you. Because sugar doesn't dissolve well into the tea when the tea is cold, right? If you heat it up, that helps facilitate it. So when they make the tea, if they add the sugar, then it's nicely dissolved. Otherwise, when you get it, when it's cold, the sugar just goes to the bottom, right? It doesn't dissolve very well, right? Not to the level of sweetness that I would prefer. Okay. So when it's colorless, when it's in its oxidized state, it's soluble, which means where is it in the media? Everywhere, right? And that's what we want, right? We want the TCC everywhere in the media. So no matter where you inoculate, it's going to come in contact with this reagent, correct? But when it becomes reduced by the organism growing, it turns red and becomes insoluble, which means now, what does insoluble mean? It's not going to dissolve into solutions. It's going to stay right where it is. And that's important, right? Because if we turned it red and it was still soluble, it would go everywhere, even where the bacteria aren't. We need it right next to the bacteria because we're using it to show us where the growth is, right? Okay, so that's motility, and we've already recorded our results for it. Now FTM and anaerobic jar. Both tests, right, determine oxygen requirements of the bacteria. My next 7 slides are basically a review of metabolism, which you're supposed to know from biology class. So what I'm going to do so that we can watch our slides, look at our slides today and save time, is I'm going to kind of skip over this part of the lecture, but I'm going to post the recording from my other class for you guys. For some of you guys that need to review metabolism before we talk about it even more next week. Okay? Okay, so now we're going to get to the terminology that you guys need to know for this course. So, can the organism tolerate oxygen, right? So we talk about aerotolerance, right? Does it have the ability, or is it unable to tolerate oxygen? Right? And this will affect where it can grow, right? If it can't tolerate oxygen, then it can't grow in the presence of oxygen. So we have about 5 different classifications that we put organisms in based on their tolerances and uses of oxygen for their metabolism. So strict or obligate aerobes require oxygen for growth, can only grow in the presence of oxygen. So that was you guys whose organisms only grew at the top of the tube where the oxygen was. They're doing what we call aerobic respiration, right? They're using oxygen as their final electron acceptor. Strict obligate anaerobes do not require oxygen for their metabolism, right? They're doing anaerobic respiration or fermentation. And in fact, can they grow in the presence of oxygen? No, it's lethal to them. So we don't have any organisms that are strict anaerobes in this lab because, again, it's too hard to maintain that oxygen-free environment, right? If this lab was free of oxygen, we'd all be dead, right? We're aerobes. So it's really hard to grow them, right? So we don't mess with them. I don't have time. Babysit those babies, right? We've tried several times. We can't keep them happy. So we don't have any examples of that for you. Microaerophiles. These are another problem for us, right? Because micro means what? Small. And so how much oxygen do they need? Small amounts, right? And again, we wouldn't be happy if there was just a little bit of oxygen in here. We'd all be passing out. So we don't work with these guys because, again, they need small amounts of oxygen to keep them happy. But they have to have the oxygen. They're still doing aerobic respiration. So you got to give them some. You got to give them enough to survive. Capnophiles are a special type of microaerophile, which, again, they need small amounts of oxygen, but they also need elevated levels of carbon dioxide. Again, we wouldn't be too happy in that environment. Facultative anaerobes, really easy to keep these guys happy, right? Because facultative means the ability to change. They're an anaerobe, which means can they grow without oxygen? Yeah, anaerobic means without oxygen. So they can do anaerobic respiration or fermentation. But they can change, which means when oxygen is available, they do have the ability to do aerobic respiration. And in fact, you generate more energy with aerobic respiration than you do with any other type of respiration. So they grow really well in the presence of oxygen. So that's why in that tube that I told you, I said, oh, that's definitely a facultative anaerobe, because it was growing very abundantly in the presence of oxygen, and its growth was decreased in the non-oxygen anaerobic zone. So a lot of you guys have facultative anaerobes. And then one of you guys has our aerotolerant anaerobe. So how is it growing? How is it respiring? Anaerobe. They're an anaerobe, right? No oxygen. So they're doing probably fermentation. Most of them are fermenters, right? Which doesn't require oxygen. But if oxygen's present, will it kill them? No. Aerotolerant implies that they can tolerate oxygen. But do they use it? No. So their growth pattern isn't affected by the presence of oxygen, unlike your facultative anaerobes where their growth will increase in the presence of oxygen. So if you're in my lecture class, you know I love charts, right? They're a good way to organize information. So to review some of the important parts of the terminology. Can an organism use oxygen, or more importantly, does it require it? Do we have to provide it in order for it to grow? Can it grow in the presence of it? In other words, is oxygen toxic for them? Will it kill them? Can they grow without it? Right? Can we have them grow without oxygen? So a strict aerobe, does it require oxygen? Absolutely. Can it grow in the presence? Yeah, it kind of has to. Oops, I went the wrong way. Okay, so it requires oxygen. Anaerobes, definitely not, right? Anaerobe means I don't use oxygen. Microaerophiles, yes, but little bit, right? Facultative anaerobes, they don't require it, right? They don't require it. Good, you guys didn't get tricked. Aerotolerant anaerobes, do they require oxygen? No. Okay, so obligate aerobes, we already said, right, that they need oxygen, they got to have oxygen. Anaerobes, can they grow in the presence of oxygen? Strict anaerobes. No, oxygen will kill them. It's toxic. Microaerophiles, can they grow in the presence of oxygen? Yeah, if it's a little bit of oxygen. Facultative anaerobes, can they grow in the presence of oxygen? Yes, and abundantly at that. Aerotolerant anaerobes, can they grow in the presence of oxygen? Yes. Can aerobes grow without oxygen? No. The only ones that we're going to say yes to are the ones that have what in their name? Anaerobe. So can a strict anaerobe? Yes. Can a microaerophile? No. Can a facultative anaerobe? Yes. Can an aerotolerant anaerobe? Yes. Okay, so we have our thioglycollate media, and you'll notice there's an uninoculated control in your guys' rack. Right? It's labeled uninoculated control, and it has FTM. I want you guys to go ahead and pick up that tube and hold it up to the light. You see the pink layer? Would you inoculate that tube to test your unknown? No, why not? There's too much oxygen in that tube. Did you see how thick that pink band is? Right? Those are from yesterday. So they're too old. They're too old to use. What we do is we will boil them for 5 minutes to drive out all the oxygen and then let them sit and have the oxygen come back in so that you get the desired very thin layer of pink. Does this mean this is the only place the oxygen is? No, it does diffuse a little bit lower. This is where the highest concentration is, right? But by the time you get to the bottom of the tube, there shouldn't be any oxygen if you have just a thin layer of pink at the top, okay? So let's talk about the architecture of these tubes. So why is there a little bit of agar, right? When you guys inoculated, there was a little bit of resistance. You can see in that one tube how the growth was like suspended, right? So, but why? Other than, you know, it really helps us visualize where the growth is, right, if it's suspended in a little bit of agar. But what's the main reason that there's agar in that tube? Yes, to slow the diffusion of the oxygen into those tubes. And then stabilize growth as well. In the media, and in the name, right, thioglycollate, sodium thioglycolate and L-cytosine, an amino acid, are used in this media. What do they help us do? What do we not want in these tubes? We don't want oxygen. So it binds with the oxygen, or it, what's the word? Here, I'll cheat. It donates electrons to the oxygen, right? So the oxygen becomes reduced, right? Which means it gains electrons and forms water. So we're removing oxygen by turning it into water. Make sense? So in chemistry class, this is a lifesaver even to this day. I learned the saying oil rig. Oxidation is the loss of electrons. Reduction is the gain of electrons. Yeah. It saved me all my life. Right? There are other sayings out there, but especially living in Louisiana, no one forgets about oil rig, right? That one's easy peasy, right? Okay, make sense? Okay. So what color is the resorrin in the presence of oxygen? It's pink, right? So that's why we know that's where there's a lot of oxygen, right? Because that reagent is also in this media and turns pink in the presence of oxygen. Is it oxidized or reduced? It's oxidized, which means it loses electrons, right? Loses electrons. Oxidation is the loss of electrons. Why do we need this? Why do we need that pink layer?
It's an indicator.
It's an indicator, right? Because can we see molecular oxygen with our eyes? No, right? And is it important for this test to know where the oxygen is? Yeah, because again, you wouldn't want to use that media. It's too old. But how would you know? Right? You wouldn't unless we labeled it with a date or something. But even then, you couldn't be 100% sure it's good. Right? So yeah, it's a very important indicator. So why do we want, or what is more desirable, thick or thin? Thin, right? And that, again, means that only a small amount of oxygen has diffused back into this tube. Remember, these aren't sealed cap tubes that we're using, right? Oxygen continues to diffuse. That's why, again, even though I refrigerated, right, especially yours, right, it grew really far down into her tube because the oxygen kept diffusing in. Right? I tried to slow the growth as best I could by putting it in the refrigerator. But typically you would read these the next day. Right? You'd read these the very next day. And we refrigerated them the very next day, but that was 2 weeks ago. So, we look for different growth patterns, and this is going to help tell us, right, which classification your organism would be. And again, because of the age of the media and such, we really can't tell the difference between facultative and anaerobic, but there's another test that's going to clue us in later, and that has to do with what level of protection do they have against oxygen and what is necessary. So our obligate aerobes, where are they going to grow in our tubes? Which tube represents their growth pattern? Yeah, number 1. Microaerophiles? 4, because they're going to be, in the less oxygen, not the high oxygen zone. Obligate anaerobes? 2. They're only going to be in the no oxygen zone. Aerotolerant anaerobes? This one's 5, because remember, they're not affected by oxygen, so notice the growth is uniform. Facultative anaerobes is our 3, which most of you guys have, right, but we really can't see it. They're going to have the really thick layer of growth, right, because they can grow faster in the presence of oxygen. And they're still going to grow without oxygen, just not as abundantly. So these are beautiful pictures from your book, right? So which one is the strict aerobe? D. D. See that? Which is the strict anaerobe? C. C. See how the pink layer, no growth, and the non-pink layer, the anaerobic layer, has growth. Microaerophile, E. So notice again, not in the pink layer, but just below it. But just below it, doesn't extend down the tube. Facultative anaerobe, B. Again, abundant growth in that oxygen layer, but still growing anaerobically. And then our aerotolerant is definitely A, with the uniform growth throughout. So, right, of course. So, why is it important that the media be fresh? What are we trying to grow? Anaerobes. Or know if your organism can grow anaerobic. So again, we don't want a lot of oxygen in the tube. So we want to use fresh media, right, so that our anaerobes can grow. Would it affect our other organisms that utilize oxygen? No, they would be happy because there's plenty of oxygen, but our results would be inaccurate, right? Because we wouldn't know that it was an aerobe versus an anaerobe if the oxygen had diffused through most of the tube. So Anaerobic jar. So methylene blue strips are commonly used, or in the case of my gas packs, we use, it comes with a methylene blue tablet. So it has methylene blue in it. This one's really getting old, so you really can't see the blue tablet all that well. But under anaerobic conditions, what color does that tablet become? It'll become colorless, right? Again, can we see if this jar is anaerobic or not? No, not with our eyes. We can't see molecular oxygen, so we need an indicator. In this case, it's methylene blue. Probably need to open another packet so we can get a nice blue. It's my baby blue tablet now. All right, in an aerobic environment, though, it's what color? Blue. Remember, we've used methylene blue dye, right? Already. You know that under the conditions of this lab, it's blue. This one is definitely clear, right? See that tablet? Yeah. No. This one's very good, right? Because this chamber at this moment in time when I took the picture was what? It was anaerobic. I succeeded. Yeah. It worked. This one was left out on the tabletop. So notice the tablet is blue, right? Which means obviously this room, which we know, is aerobic. Otherwise, like I said, we'd all be passing out. That packet comes wrapped in foil, right? So I've got some of them in here. That's not the issue. It's the jars. It's the issue. Once you open this up and expose it to the air, when you stick it in the jar, and you have to put 3 for this size jar, a chemical reaction happens that releases hydrogen and carbon dioxide gas. The hydrogen combines with the oxygen to form water. That's how we get rid of the oxygen. That's one of my jars. What do you see here? Condensation, because what's forming? Water. So I know, again, it's working, right? And if I looked at my tablet, hopefully once the condensation clears, it should be white or colorless. So if the environment is anaerobic inside the jar, how would it affect the growth of these organisms? So if we put known anaerobes in this jar, and we make it an anaerobic environment, they're going to be like, woohoo, party time, happy, they're going to grow, right? What if I stick an aerobe in this jar and make it anaerobic? I'm going to kill it, right? It's going to go, you killed me. What if we stick a facultative anaerobe in here? It'll grow, but slowly, right? Because it's going to have to resort to fermentation probably, which is not as much energy. Make sense? Okay. So if the environment is aerobic, so my jar springs a leak, how am I going to know that happened? My tablet's going to be blue, right? But also if I put known anaerobes in this jar, right, when I open up the jar, they're going to be what? They're going to be dead. And the aerobes are going to be growing, right? And that's not what I expect. Are the facultative anaerobes going to help me out here? Maybe if I notice their growth is what? More than it should have been under anaerobic conditions. They're growing abundantly. So in the experiment, what we would do is put organisms inside and outside the jar. Known aerobes and anaerobes, right, as our controls. And we'd put your unknown in here. So what is the environment inside the jar? What's this jar called? Anaerobic jar, right? Is oxygen present? No. Outside the jar? Aerobic. Is oxygen present? I hope so. Okay, so these organisms are our controls. But what are they a control for? So if Pseudomonas has no growth in the anaerobic conditions inside the jar, and it grows outside, this is our aerobe, right? Because it only grows under aerobic conditions. Clostridium sporogenes grew inside the jar, but not outside. Anaerobe. Again, I don't mess with this baby. It's too hard to keep it happy. And by its name, guess what it does? Forms spores. Staphylococcus aureus grows inside the jar and grows really well outside the jar. This is our facultative anaerobe. So, Pseudomonas, does it require oxygen for growth? When we stuck it in the jar, what happened? It died, right? Remember, this is our obligate aerobe, so yes, it needs it. It won't grow without it. Clostridium sporogenes is our anaerobe, right? So does it require oxygen? No, in fact, oxygen would do what? It would kill it. Staphylococcus aureus is our facultative anaerobe, right? So does it require oxygen for growth? No. But how does oxygen affect its growth? It'll grow abundantly. So here's an experiment. Well, here's the one in your book, right? So if it grows under both conditions, it's facultative. If it grows only under anaerobic conditions, it's an anaerobe. If it grows only under aerobic conditions, it's an aerobe. So they just spot inoculated one plate, right, with 3 different organisms. Here we inoculated 2 sets of plates, 3 different organisms, put them in the jar and outside the jar, and this is their growth. So you can see these guys grew in both conditions, both E. coli and Staph aureus. So they're what? Facultative. Pseudomonas, it's hard to see its growth, but it produces a green pigmentation under certain conditions. So you see this green pigmentation, it grew under aerobic conditions outside the jar. But you don't see any pigment inside, right? It was killed. There was no growth. So this is a aerobe. Right? So it's an aerobe. Our Staph and our E. coli are both facultative anaerobes. An alternative to the anaerobic jar would be a candle jar, where you take a big jar, you stick a candle in it, you light it, you put your cultures in, and you close it up. What happens to the oxygen level in this? It decreases as the candle is burning. So who would be really happy in here? Not really anaerobes, because it's not going to remove all the oxygen. Microaerophiles. And a specific subset you just mentioned, the capnophiles, because what are capnophiles like that's going to increase in this as the candle burns? The CO2. Right? So those guys, which are a type of microaerophile, you're not going to eliminate all the oxygen, but most of it will be. And that should be it for this one. Yep. All right.